Hello, everyone. Thank you for joining us for this behind the scenes at a soil lab webinar. My name is Catherine Garcia, and I will be the moderator for the webinar. As everyone's trickling in, we're going to go through a few housekeeping items. First and foremost, as a reminder, this presentation can be used for CCA continuing education credits using the QR code at the end of our webinar. In 1959, AgSource was formed to serve Wisconsin dairy farmers. What began as a regional dairy records processing cooperative has now grown into the AgSource of today, serving industries that span the entire agricultural ecosystem. From cutting-edge soil, plant tissue, and water testing to agronomic consulting and full-service DHI testing, AgSource at its core is a source of truth. One test at a time, we help our clients make the best decisions, always grounded in data. I'm now excited to introduce our presenter, Jim Friedericks. Dr. Jim Friedericks holds the position of Outreach and Education Advisor for AgSource Laboratories, coordinating the education programs of AgSource Laboratories and supplying technical information to industry and agronomic consultants throughout the country. Soils and environmental sciences are central interests in Jim's work and educational experience. He received his BS in agronomy from University of Kentucky and a PhD in agronomy focused in soil fertility from Virginia Tech. Jim has over 25 years experience in soil testing, joining AgSource team in 2012. Also joining Jim for our Q&A portion of our webinar is Kevin Klink, Director of Agronomy Laboratory Operations at AgSource. I'm now excited to turn it over to Jim to get started with our presentation.
Thank you, Kate. It's good to be here this morning with everyone. As Kate said, I'm in charge of outreach and education with AgSource, and I also work in the area of research and development. And from that perspective, talking a lot about the quality of what goes on in a soil testing lab, and from my experience with AgSource as well as other laboratories over time. What I'm going to say today is not just focused on AgSource laboratories, but is in general applicable to the soil testing industry as a whole. Thanks for joining us today. As we get started thinking about what goes on behind the scenes at a soil testing lab, I just want to take the perspective of what do we think about when we think about service providers that we use. What sort of things do we anticipate getting from a service provider? Well, first of all, we want someone who's trustworthy. So obviously you want your bank to be trustworthy, but you also expect your weatherman to be trustworthy and give you reliable information. You want the service to provide affordable service to you, whether it's the education that you pay for, or maybe getting your car serviced. You want it to be something that's value for a dollar that you're paying for. And then it needs to be applicable information or applicable service to you as well, whether that's safety warnings that come with packaging and instruments that you buy, or maybe it's a health provider providing you applicable information that helps you manage your health. So those are the sort of things that we have in mind when we think about service provider. When we think about soil testing as a service, what are those same things in that kind of context? First of all, we want consistent quality of data. Somebody who uses soil test information wants to make sure that that data is good, it's reliable, it's can be counted on. Then obviously we want it to be reasonably priced. There's a range of prices that you can pay for soil testing service, and from that, you want to make sure that what you're getting is a good value as well. And another concern for soil testing service is the turnaround time of the information. Once you send the sample in, how quickly can you expect to get the data back? All of those interact, obviously where they overlap, that's where the sweet spot is, so to speak, and that's where the data is relevant. It's good quality, it's reasonably priced, and it gets to you when you want it and in good effect. Looking at that same relationship in this way, the triangle of those three criteria: we want high quality data at affordable price and high speed or reliably good turnaround times. They have good speed. These three criteria are often in conflict with each other. You can emphasize one or two, and that will de-emphasize one of the other ones. For example, you can emphasize speed and quality in a soil testing program, getting the samples back quickly and making sure that the data is really good quality data, but you may not be able to do that at a low cost. And so the price characteristic goes up. When you take that, those two relationships and prioritize them, if you prioritize the quality and the price, that actually may slow down the analysis, where you spend more time looking at each sample individually, making sure that the quality is good, but that will cost you somewhat and may slow down the analysis. And then the other combination, which is a fast turnaround time and a low price, unfortunately, what you jeopardize with that combination is the quality of the data. And we want to make sure that as a lab, the quality of the data is good as well. So it's not easy to balance all these things, but that is the priority that we do try to take in a soil testing program, whether you're a commercial lab or a university lab. High quality data at a reasonable price and in a turnaround time that's going to get everything back to you when you want it and when you need it. What that provides for you then as a user is accurate data that's appropriate, meaning that it's there when you want it. It gives you information that you can count on, that you need. Is it the right soil test for the questions that you want to answer? And are the ranges that are being reported to you pertinent, are they appropriate for that? Is it timely? Is it coming back to you when you need it? Does it address the need that you have in that it's pertinent to your requirements right now? It's not always useful to have old data that's been sampled a long time ago when your questions change over time. And so it may be more important to get samples back right before you're making decisions. And then, is it affordable and applicable? Of course, the cost is an important characteristic, but is it applicable? Can you take that information and apply it in a situation where it will make a good management decision for you and give you an outcome that you're trying to achieve? So high quality service and high quality data from a laboratory actually starts before the sample gets to the laboratory. And I'm going to spend just a few minutes talking about the sampling strategy, what goes into collecting a good sample, and how that really ties in with the analysis and getting pertinent data back to you. Often in a laboratory, a question will come up about results that somebody gets back, and one of the first questions that needs to be resolved is, was the sample taken as it was anticipated from the location where it's supposed to be, correct depth, and all those characteristics. And so getting the right sample is a real key to getting a good analysis in the laboratory. When you walk into a field, there's always going to be variability in that field. The image on the right is the soil survey image of a given field. And you can see there's a lot of variability from north to south in that field. And what is dominant? Well, it's hard to say. Maybe that dark brown is the dominant soil, but there's a lot of other soils that are present in that. And each one of those will have different characteristics when you sample it. Excuse me, to go out and sample that, you could take one sample, which would be made up of cores collected all over that field, and that will give you an average of the entire field. That gives you one value, doesn't really help you understand all the variability that's there. Or you could divide that into multiple areas, such as with the image on the left, the graphic image, where we do a grid sampling of that field and divide that sample up. In this case, it's divided up into 15 relatively equivalent areas, and a sample is collected from each one of those areas. That will give you 15 observations of that field and give you a better indication of the variability. If you increase that to 60 by dividing each of those into 4, you get even more information about the variability of that field. And so the smaller the grid size, obviously, the better your resolution and understanding of that variability, which will all go into helping you manage that field for the optimum outcome that you have in mind. So how do we know what is the right sample to collect? Take this image here that shows the variability in a given field that's just say represents an acre. And you could go out and sample all those different blocks and be able to reproduce this map, or you could sample it in quarters, or you could sample it in various ways just to try to get that representative representation of that variability in that field. Any spot that you pick, if you just decide to pick one spot, you can see that you may have one area highlighted, such as the green area, or you may end up in what's the red area, or you may end up in a spot that includes all those areas. So it's important to know what you're sampling and what you're trying to achieve with that sample. Typical grid sampling, rather than trying to get an average of the whole area, typically what happens is a location is picked within that block, and then from year to year, or every time that site is sampled, you go back to that same location and resample that same location. That way, any management practice that you apply to that whole area, that acre in this case, would be reflected in the changes that happen in that location. And so you're monitoring a change, but you're not monitoring in the entire area. So that's the philosophy behind grid sampling a point versus trying to get the average of the whole location within the grid or within the field. Other characteristics to keep in mind: what is the right depth of soil that you want to collect? And that depends on a couple of things. First of all, is the soil going to be plowed, or are you practicing no-till? And here I'm primarily thinking of agricultural, but if you're thinking of sampling a lawn, again, a shallower sample might be appropriate for 4 inches in a lawn situation, or 4 inches in a no-till situation will give you the distribution of fertility within that area of soil or depth of soil that you can influence with the surface application. If you're tilling and turning over the soil with different tillage operations, 6 inches or 8 inches is maybe more appropriate. If you're looking for nitrogen accumulation in the topsoil before applying nitrogen mid-season for a corn crop, your depth of sampling would be probably 12 inches. So just have that information in mind and keep that in mind when you're planning your sampling program. And then again, how many cores is appropriate? It's more is better. Obviously, you want if this picture represents an acre and you wanted to know exactly how much variability that was involved in that, you'd sample every one of those little blocks to get the actual variability. But if you want to get an average of that, how many cores would it take to get something that represents that average? This graph is an image of some research that was done in Iowa by Dr. Malorino a number of years ago, in which he took an acre of soil, sort of depicted by this picture, and sampled it 45 times. And what I mean by that is he took 45 cores and he analyzed each core for phosphorus content, and then he mathematically determined what was the median soil test value of that area by compiling all those cores. And he came up with 19 being the average or median parts per million of that area. So if we take 19 ppm to be the true value of phosphorus in this example, how many cores does it take to be able to predict that with the confidence that you want to be able to have in that prediction? Well, to get it exactly right, you want to do 45. But if you back off from that, well, what about if you only did 10 cores? And at 10 cores, you can see the number represented on the bottom. And then that where you go up and intersect with the curve, you see that number represents about a confidence interval of about 4 parts per million, meaning that if 19 is the true value, you know it's going to be somewhere between 15 and 23 ppm, and that's about a 78% confidence interval. Is that good enough for you? That's pretty wide. If you wanted to get a little bit more precise, you could up the number to 15 cores, and that would give you a confidence interval of just over 3 ppm, or somewhere between 16 and 21. Excuse me, and that's probably more comfortable. And usually samplers will take 10 to 15 cores per sample. So example again, if this is a grid of 1 acre or 2.5 acres, and you decide that spot that's circled is where you want to collect your sample every year, then typically 10 to 15 cores collected in that location will give you a high quality value that you have about 80 to 85% confidence in. And you can go back to that same location and collect 10 cores again next time and be reasonably sure that the value you get represents within that confidence interval what you're anticipating it will be. Whatever your plan is, stick to it. Create a plan before you go into the field. Have that in mind. Get your grids, locations selected, and then when you go out there, make sure you're going to the right location. You're sampling to the correct depth. If you're intending to collect a 6-inch sample and you only collect a 4-inch sample, then you're biasing the results because you're not collecting the true depth that you have in mind. So make sure that samples are always the right depth. Collect the number of cores that you want to have and to represent your confidence that you want to have in the sample, and then make sure your samples are clearly identified. Typically, excuse me, 10 cores will usually fit in a bag, just like that bag in the picture, and that entire sample core then will be sent to the laboratory and used for analysis. And unlike the picture, make sure you have clear identification written on the bag before you seal it up and send it off to the laboratory. Samples that come to the laboratory then from the field start to be treated by the lab with a program that maintains a high integrity of the sample, and it's consistently carried out every time for every sample, so that we know that with a high degree of confidence that everything is done the same way, and we know that sample is the sample that we're working with. The samples typically arrive at the laboratory either through the postal service or a delivery package service. Some laboratories use routes, and they will go out and collect samples from samplers or from companies that submit large volumes of samples and get them to the lab. Usually try to get everything into the lab early in the day and start the process of logging in those samples and getting them into the system. All that information that's submitted on the information sheet is typed into a computer program, which is the lab information management system, and that information is married with the analytical data later by keeping track of the unique sample ID that's assigned to every sample, and then all that information goes into generating the reports. When the sample is received, all the bags are lined up, making sure that we have all the samples, all the physical bags that are indicated on the information sheet, and they're placed in order, and then assigned a unique number, as well as a location in a tray. And almost every laboratory will follow the same practice, where samples are given a specific spot in a tray, and then from that point on, it's that location in that tray that identifies the sample for the laboratory, but that unique login number is what's used to marry the data to the information sheets and to finally on the reports that go back out. Samples that come into the lab are spread out in trays to dry, and then those trays are loaded into a drying room. Temperature of about 105 degrees Fahrenheit is enough to just raise the temperature of the air and start to remove the moisture from the soil. That air is replaced with dry air, and within 8 hours, typically you can start to process the samples by taking them out of the drying room and grinding them. So it's a fast-moving process, trying to move a lot of samples through the drying room so that they can be prepared for analysis, typically the next day after the samples are received, or later that evening that they're received. The samples, when they're dried, are taken from that drying tray, poured into the top of a mill. And this grinder is a chamber with rotating flails inside the chamber that breaks up the aggregates of that dried soil sample and separates all those aggregates into smaller particles. That then is dropped through a screen, and whatever passes through that 2 millimeter screen is what's used for the analysis. Anything larger than that is discarded, including roots and cobs from the corn plants or whatever stones that may be in the sample. That's all separated out, and what we go forward with then is dry soil material. But there is an exception to this. Some laboratories, and particularly here in Iowa, where I am, we offer a moist soil extraction process where the soil is not ground, but we take the moist soil right out of the field as it comes to us in the bag, and weigh a portion of that and mix it with water, and then suspend that soil in the water, and take an aliquot of that suspension out, and then carry that forward into the extraction process. That aliquot that we take out is calibrated to be the same amount of soil that we would use if we did a dry extraction or a dry soil measurement. The reason behind this moist extraction, and it's only used, particularly used in potassium analysis, is that sometimes that drying process for some soils will change the availability of potassium in the soil. And so there are different ways of interpreting that based on whether it's a dry analysis or a moist analysis. But this is just an exception. Otherwise, everything typically goes through that drying process that I just described. All those steps that I've described so far are pretty consistent, regardless of where you are in the country, what laboratory you're using. Generally, the same methods are applied in terms of sampling and receiving and drying and grinding samples. And these methods are methods that have been in place for many, many years. And then next, I'll start talking about the actual soil testing itself and the methodology that's used to extract the nutrients and to analyze the relationship between nutrients in the field and the soil and the plants that are growing them, and the ability of the plant to achieve the yield goals that are desirable. These methods are based on research that have been done primarily in universities starting in the 40s. And then as fertilizer industry started to make fertilizer more available, these methods have become more and more important in terms of learning how to manage the fertility of the soil and then make the nutrient applications that are required to achieve those responses. So it all goes back to university research, and because every state has a university that perform this, those differences develop because of the regional nature of the United States and the specifics of those universities where they are. That research has been shared primarily in states that are nearby, have found synergies in sharing that information and cooperating together in working groups so that the soil testing methods are actually developed with specific areas in mind. So AgSource laboratories, our laboratories are located in the north central region of the United States, which if you see the map on the image on the right, is from Kansas and Missouri all the way up north to North Dakota and far east as Ohio. That's the north central region. All of us in this region follow guidelines that are put together by this regional working group. The methods that we follow are set out in these procedures, and we follow those methods as they're laid out, so that we can both share information, share results, as well as transfer those samples back and forth and that information back and forth. There's a Western group, there's a Southern group, there's a Northeast United States group, and there's a group in Canada. So these regional areas are important. If, for example, our laboratory in Nebraska receives samples from farther west, they use methods that are appropriate for those samples in the Western group, whereas samples in Wisconsin use methods that are appropriate for the North Central region and particularly for Wisconsin. There are a lot of laboratories in the United States and Canada and Mexico, and just a quick look at those that participate in one of these testing proficiency programs. There's 130 labs in that. So the North American Proficiency Testing Program, the one I just referenced, sends samples out to laboratories all around the United States as well as Canada and Mexico. And we use them, we being a laboratory, use them to help us evaluate our proficiency, our ability to perform an analysis and make sure we're doing a good job. There are several of these proficiency programs. Agricultural Laboratory Proficiency Program is another one, which is national as well as international. If we're involved in manure testing, there's a manure proficiency program that also certifies laboratories. ALTA is one that started in Illinois, but it's now spreading into more and more states to certify labs and help labs meet those proficiency programs. And then additionally, as I mentioned, in Wisconsin, there are specific certification programs because they have specific tests that they perform. And so the Wisconsin Department of Agriculture has their certification program, and Iowa has one as well as other states do too. All those programs are the background for making sure that both the lab is certified, but what goes into that certification is a good quality assurance program for the laboratory. How well does a lab perform what it says it's going to do, and is it able to meet those proficiency characteristics or requirements every time, quarter by quarter by quarter on an annual basis, and then to receive certification on an annual basis. So every laboratory has to set up certified operating procedures so that they're following these procedures every time they do an analysis. And then part of that assurance program is making sure that the chemicals they use are certified chemicals from a reputable distributor, as well as maintaining good instrumentation and calibration of those instruments. All that is documented by the laboratory as part of their analytical assurance programs. To review the background of this, and then I'll talk more about some of the methodology, but assuring the quality of the samples we get. The samples are received, they're logged in, they're ordered. We insert a quality control sample into every tray. I'll talk about that more in a minute. The samples are then dried and ground, and then we start moving on to the actual analysis where we scoop the sample for the dry sample, and then we also have a quality control sample that we include in every tray. If you look at the bottom of the picture there's that orange block in one of those cups or boxes. That's a marker so that when we go through and start scooping the sample, when the technician gets to the box with one of those markers in, rather than scoop what's in a box, they scoop from a bulk soil sample. And that bulk soil sample is a sample that's been tested over and over again. And we use that just to monitor the quality of the analysis as we go through, make sure that that analysis is meeting the expectations of what we're doing. So a scoop is transferred from the dry soil, as you can see that fine powder, into another cup. And then from there, it's taken for processing. Samples that are analyzed for phosphorus or potassium or any of those other elements are typically extracted with a solution. And there's different solutions that are used for different extractions. The solution is shaken for a prescribed amount of time, filtered out, and then when that filtrate is collected in small tubes, which you can see some on the instrument there, and those racks of test tubes. And those test tubes are then analyzed for nutrient concentration. So in this case, the instrument you see there is an ICP, and the solutions are being analyzed for potassium, calcium, magnesium, and sodium. You can analyze all those nutrients on one sip from one sample, and so you can get a multiple number of analytes with one analysis of an instrument. And the technician then is responsible for calibrating the machine, setting the samples in place, checking those quality control samples every so often, make sure that they're being falling within the ranges that we anticipate, and then passing that information on to the computer for incorporation with the rest of the data. If the different analyses have different kinds of instrumentation that they use, this one is for colorimetric analysis, and we use it for phosphorus determinations. You can see in the background of the tubes, there's a row of 6 tubes in a row, and then one sitting off by itself. Those 6 tubes are the standards. So before a tray is analyzed, the instrument is calibrated with a set of standards, which includes a blank. And then that single tube by itself is a check sample. So after the calibration, the check sample is read, make sure that it's reading what you expect it to read, and then the samples can start to be analyzed. And typical practice would be after every 10 or 20 samples that are analyzed, you would go back and read that check sample. And if it's still within the expected norms, then the instrument is okay to start reading the next samples. If it fails, then the program is set up so that the instrument will go back and recalibrate and reanalyze the samples after it passes the calibration. So the technician then, in this case, is monitoring it, but the instrument is set up so that it can actually do the calibration itself. And all the technician needs to do is make sure that the instrument is running as expected. For pH analysis, we mix the soil with water. You typically a 1-to-1 or a 1-to-2 soil to water ratio. Stir that up and then dip some electrodes into that suspension and read the pH of that with a pH meter. That is used for water pH. For lime requirements, which is often called a buffer pH, we add a buffer solution to that mixture and stir it up again, and then read the change in that buffer pH solution. And that's what we use to estimate or to calculate how much lime is required to change the pH of that soil. So here again, you can see an automated system where we can read multiple samples at a time. For organic matter, the samples are scooped into these crucibles, and a crucible is heated up in a furnace so that the sample is actually burned. It's a controlled temperature. And what we're doing is burning the organic carbon out of the sample, but not oxidizing the carbonates, and then reduce the temperature back to oven dry, which would be on 100 degrees centigrade, and record the weight change from before and after the sample was burned. That weight change is called the loss on ignition, and that's what you use to calculate the organic matter content in soils. So these are routine things that I just showed you. Every lab will do all of those tests. There's also other tests that are offered that require different programs, but by running all these routine tests with the same methodology and by as much automation as we can, quality is assured by just making sure that everything is done the same way, optimizing the throughput by just managing the amount of time that it takes to read each sample, and then making sure that the instruments are calibrated and performing as they are expected, and making sure, or having the computer recalibrate the instrument as needed. So a lot can be done automatically or electronically, and the technician is just monitoring what's going on. The quality control samples that I mentioned are placed in every tray. Typically 5 to 9 or 10% of the samples in a tray are those QC samples or quality control samples. And as I said, the technician is able to look at those and monitor that to give them a real-time evaluation of the performance of that tray. Is extraction going as expected? Is there a problem with the extraction? Is there maybe, if it's out of order, the random location of that quality control help you identify if the tray is the correct tray. And then all that is used by the quality assurance manager to make sure that everything is done according to our procedures, so that we have a good quality data that comes out. And what are we looking for? We want to make sure that the data is both accurate and true. Is it accurate in terms of this graph? You can see accuracy is the vertical axis and precision being the horizontal axis. So a sample can be accurate on average over on those left-hand two circles, where the circle is a little bit tighter, you have a better precision. Whereas in the bottom, it's accurate and it's not very precise at all. The sample points are very well distributed and kind of randomly scattered. Or you can be very precise, meaning that you have all your results in a very tight cluster, but that cluster can be in the wrong place, which is the bottom right illustration. And that's a bias. So it's very precise, but it's not right. It's not true. And so what we're trying to achieve is both a high degree of precision by calibrating the machine so that we get a very close tolerance of the range of values, as well as a high degree of accuracy, meaning that the scooping, the drying, the grinding, all those things have been done carefully so that the sample is well homogenized and it represents what's really in that sample from one analysis to the next. As the data passes through, of course, we do use criteria to evaluate it and look at the variability of that quality control sample and see if it's, looking at the standard deviations, if it's 1 or 2 standard deviations, it is okay. If it's more than 3 standard deviations from the norm, it is excluded. And we plot that information as it goes through. This is a plot of pH data. And you can see over time, sample 1 on the left and sample 1,000 on the right, we saw some variability. We saw a few places where it was outside the range, the upper or the lower specification limits. And so those samples had to be redone, but everything else that falls within that range was acceptable and could be passed on for reporting. So every tray has those knowns. Those results are watched carefully. And these are the kinds of criteria that we use in the laboratory on a day-to-day basis. But it's also the kind of criteria that we use when we receive samples from one of those certification boards to make sure that we are performing in the expectations of the certification boards. AgSource is like any other laboratory. We participate in several of these certification organizations: the NAPT, the ALP, ALTA, and the Minnesota Department of Agriculture, which runs the manure testing program. All those are used by our laboratory to help us be sure that we're doing a good job, but also to demonstrate to the states that certify us that we are meeting certification performance criteria. Knowing that we are doing a good job with quality assurance and quality control, that provides confidence to our technicians. When they're doing the analysis, they know that they're doing a good job because they have points of reference as they do the analysis. The managers and the quality assurance managers can observe that and of course spot things that aren't being caught, but can release data with confidence. That of course is good for relationship with our clients and our growers, who can have confidence in the data that goes out. It's the cost of doing things right. This goes back to that triangle of balancing cost, speed, and accuracy. And it's one of the things that it's 10% of the samples that we run are quality control samples. So there's a cost involved, but it's the cost of doing things correctly. And it's important to maintain that balance as we go through samples as quickly as we can, but maintaining high quality and doing it in an affordable way with the programs that we have in place that speed up the analysis by handling samples in large amounts, trays of samples, and automating as much as we can. That assures that the data that goes out is accurate and it's appropriate for the region that we're serving, or the client, where the client sent samples from, and the plants, the crops, or the lawn and garden, or whatever it is that you're submitting the sample for, so that the data is obtained and you have confidence that it was handled carefully, and the results get back to the client on time so that they can use that information in a way that helps them make good management decisions. As I said, the university research is the basis of all this, and the testing methods that we use have been developed through the university trials on field, and then methods that the universities have developed over time that helps them interpret the data. So the interpretation of the results gets back to what does it mean in terms of what this soil test value means in the soil, and what does that look like in terms of the ability of the crop to yield a certain expectation. And then how do we add nutrients to the soil to achieve that kind of yield level or yield goal, and that's interpreted as fertilizer rates. The different methods, some of the different methods that are used for potassium and for phosphorus are here on this slide. Malic phosphorus and Bray 1 phosphorus are typically used on soils that are slightly acidic, and are used in the Midwest as well as in the eastern part of the United States. Olsen extraction for phosphorus are used for soils that are more basic, higher pH. And so you can see that the ranges are a little bit different. You know, what is adequate for Bray and Malic from 16 to 20 is 10 to 13 for Olsen. It's just because a different kind of solution that extracts different amounts of phosphorus. The same is true for potassium extractions. The Malic potassium or the ammonium acetate potassium, that center column, the adequate range is from 161 to 200. In Wisconsin, they use a Bray, the same Bray that's used for phosphorus is used for potassium, and the ranges there are a little different, 100 to 130 are the adequate range. And then that moist soil extraction that I mentioned, it's just because the sample is treated differently and it's not dried out, it gives you a different range of soil potassium, and those ranges are shown there. So these numbers are categorized way over on the right by very low, low, adequate, high, and very high, indicating that's kind of you don't want to be in the low range, meaning you're probably not enough fertility, and you don't want to be in the very high range, meaning you probably have too much fertility. You want to be in that adequate range. What does that mean in terms of crop growth? This graph shows the relative yield on the vertical axis and the soil test value. In this case, it's potassium, but that could be any number on the horizontal axis. And you can see, so each of those dots represents a relative growth of a crop grown in a location with a fertility soil test level at that location. So you see several, number there at 20% relative growth, and the soil test is probably 25 or 30, probably maybe 20 ppm potassium. As you go up, those numbers, as you move from 0 to 50, you see that there's a sharp increase in the relative yield of those plots until you get up to around the 100% relative growth, and you're seeing data that's anywhere from 50 up to 200, sort of a horizontal line. That means in terms of those low, very low, medium, high, and very high categories is samples that test in the very low and low range. You can, if you increase the fertility from 20 to 50, your chances of seeing a increase in yield go from you're somewhere down around 20% relative yield. You may get an 80% of relative yield just by increasing the fertility up to about 50 parts per million. But if you get above that 100 parts per million, you don't see much change in the relative yield. In fact, it's very unlikely that you'll see a response to that. And that's the probability of those responses is what's used to set those categories. So the very low and low category, there's a high probability that you'll see a response to an increase in soil fertility from one level to the next. Whereas if your fertility is in that adequate to or optimum range, which I said for phosphorus was 16 to 20 on the Bray and the Malick phosphorus, 3 years out of 4, a crop would not respond or would not be limited by fertility if it had that level of fertility in the soil. 1 year out of 4, it may respond or may be limited by fertility if everything else is really good. So that's what that 25% probability response is. If it's in the high category and very high category, very unlikely that your fertility is limiting the yield of that nutrient that you're looking at. The guidelines that are used then to make these fertilizer recommendations are let's manage the fertility of that field by replacing the nutrients that are removed when the crop is harvested. And then if the fertility is below that adequate level or that target level, then let's add enough nutrients to bring that fertility up to that level where we can manage it and maintain that, where the probability of seeing a response is minimal, 25%, maybe you want to be a bit higher than that, but somewhere in that range. And so you add enough fertilizer to achieve that level, but not so much that you're putting more on than you actually need from year to year. Managing soil pH is a little different. We don't set those same categories, but we do want to see pH managed between the pH or in the pH range that is appropriate for the crop or the plants that you're growing. Different pHs have different influences on the fertilizer availability or nutrient availability. And so a typical target would be somewhere between 6 and 7 pH, which is slightly acidic, maybe 6.5 to 7 for many crops. And you can see that because the bands are wider there, potassium is wider there, phosphates are wider there, magnesium is wider there. And so that's really kind of the optimum range where you want to try to manage fertility. If you get too acidic, then you run into toxicity problems. You get too basic, you run into unavailability of some elements, phosphorus being one of those. Nitrogen is different. We make nitrogen recommendations not based on a soil test so much as in what's expected in the crop response. And so the response curve is the same, but we don't typically look at soil test levels because nitrogen changes so much from season to season. So the university research is ongoing. As I said, the nitrogen rate studies are things that continue every year, and adjustments are made as crop varieties change, as yields tend to increase because crop varieties change. It may take more or maybe less nitrogen and other fertilities to manage those higher yields. Another area of interest that universities are looking into, as well as commercial laboratories, are the areas of soil health management, soil health assessment, and carbon sequestration and measurement of that. These are areas of ongoing research. We probably will see more publication of methods that are more universally applicable. Right now, that's still evolving, and it's not quite the same level of cooperation that we see in the fertilizer recommendations and fertility management. But it's coming. I think there's a lot of cooperation, a lot of sharing of research with soil health assessment and carbon measurement and carbon sequestration measurement. Ultimately, that's kind of what's behind the scenes in a soil lab. We want to, as an industry, as an individual laboratory, we want to make sure that we're doing a good job balancing the provision of high quality data in a timely way that's applicable and affordable to those people who send in the samples. It's going to be, is it useful for management decisions? Does it come to the grower or the user at a time where they can take that information and apply it right away? And is it going to impact the use of that location, that ground, for crop production, or maybe it's for recreation in turf grass or golf courses or something like that? Or is it, we're also stewards of the environment, we might want to make sure that our recommendations reflect that, and we recommend just the right amount of nutrient so that we're not over applying nutrients on the ground that we're managing. So, and I think I'll turn it back to Kate.
Thank you so much, Jim. We did have a few questions come in, Jim, and I don't know, I'll let you determine if you or Kevin is going to take the lead on the question. But the first question we had come in is, how do you guard against contamination in the hammer mill where the sample is ground up?
Good question. That comes up when people come into the lab and tour the lab as well. If you saw the size of the sample that we were putting in there, it's typically a pound or a pound and a half of material that is dropped into the sample, into the top of the grinder. And that entire sample is pulverized within that mill and then dropped out. So we leave it in there for long enough to break it all down and make sure it's pulverized, not pulverized in the sense of breaking the particles, but for loosening it up. And then it falls through. What typically is left behind is just a fine dust powder that clings to the plastic. We do drop it through and then open and close the chamber a couple of times with some force to make sure that there's enough banging, so to speak, to make sure anything that's adhering to the sides drops out. But the limited amount of dust that sticks to the sides of the vessel inside of the chamber is very small compared to the volume of soil that we're putting through each sample. And so we've looked at that, and the contamination is minimal.
Great, thank you. The next question, as someone who's worked during soil season, I know that there can be some changes as the season goes on from the initial reading of the elements. Is this simply due to the age of the QC soil, or due to changes in instrument readings, excuse me?
Interesting question. At first I thought you were asking about differences in the soil samples as they come in, and those spring versus fall, there are differences. But the emphasis on the changes in the QC sample. Typically, if there's a couple of ways of doing that, and we do both. One is to collect a bulk soil sample in the laboratory, and then maintain that one large sample as our QC reference. And that is something that we blend every day in something, in as sophisticated an instrument as a concrete mixer, but by blending it for half an hour each day, we make sure that it's well stirred, and then take a vessel of that and use that for the QC when we do the scooping. So that, yes, that will change over time, just because you're removing some of the soil every day, and over time it'll segregate somewhat, but we try to minimize that by blending it. The other approach is to purchase a soil from one of these certifying agencies, such as NAPT, and blending that. It's already blended before we get it. It's homogeneous, but then in the laboratory, making sure that that's mixed up, and that is used. Because it's a purchase sample, it's more expensive, so it's probably not used as often as the bulk soil, but we do use that to check every day to make sure that over time we're not drifting appreciably as well. So yes, there is some change that you would observe. It's not from the instrumentation as much as it would be from a transformation of that bulk soil over a period of time, as it gets from a full barrel to as that volume goes down in the barrel. That would be that's my observation as well. Yep.
Okay, next question. Besides the soil microbial activity test in terms of CO2 release, are you testing or planning to test the soil microbial biomass as a microbial component?
We So that's a soil health analysis, and we do measure the carbon dioxide respiration rates, or the CO2 evolution rate of the microbial biomass. I'm actually going to invite Kevin to answer this question because this is something we are interested in, we're talking about, and in terms of actually measuring biomass or diversity, that's a projection question, and he's my boss. I'm going to ask him.
Thanks, Jim. Yeah, as Jim just stated, it's certainly something we're interested in. We do not have that ability today, as Jim stated. We're looking at it as a total mass or as that release curve. But yeah, so in the future, I'd say definitely anything's possible. But for right now, and the way it looks for especially this spring and perhaps even the rest of the year, we're sticking to utilizing the CO2 release curve analysis as a component of our soil health.
Thanks, Kevin. I will add, it's sort of related. Another bioassay that we do perform in AgSource laboratories is a nematode assay, and that's that's a specific organism that we're looking at, but we do perform that as well. So, and the same criteria go with the performance of that. Thanks.
Great. And our final question we had come in, I lied, we just had one more pop up. Do you see more contamination of samples taken with an auto probe versus a manual probe crop residue and sample? Does minor corn or soybean residue confound the results?
There's probably a little bit, because the manual probe, the sampler is intentionally avoiding a coring through a crop stalk or something like that. So there is less of that residue that gets brought into the lab from a manual operation than a robotic operation. But regardless, the grinding and screening process that we use would screen out the residue anyways. And so it's really not a major concern unless it impacts the total volume of the sample that we get. And that would be, a smaller volume of soil would be a problem, but otherwise it's not a major concern.
Thank you. Is there any effort to get all the states to move toward a common extractant instead of everyone doing something a little different?
I don't think so. There is emphasis, or there is interest in building a database so that the different extractions, the different values can be compared on a comparable basis. So that would be probably relativizing the numbers in some way. But because soils, because regions of the United States have very different characteristics, an extractant from that's appropriate for the Southeast United States, where the soils are acidic and highly weathered, is not as appropriate for the Western states, where the soils are arid and basic and not weathered as much. And so you have very different confounding characteristics that influence the availability of nutrients. And so the extractions are designed to give information that's more appropriate for each location. On a smaller region, between regionalizing or within a region, trying to normalize the methods used within the North Central region, for example, there is some interest in that. For example, Bray K being used in Wisconsin and Malick K being used in Iowa is confusing. And so there is some interest in using one extraction that would be applicable to that region, but not too far beyond that region. Thank you. That's a good question.
And my last question, until they make a liar of me yet again, could you let me know what you think the advantages and disadvantages of drying the soil at 105 degrees as opposed to 95 degrees?
There's minimal difference. 95 degrees would potentially take longer just because it's cooler, but as long as you're removing the moisture out of the air, it should dry just fine. The reason we stop at 105 degrees is we found that over 110, that's where you start to see the differences in the mineral, the clay, transformations in the clay that change that potassium availability. And so managing it below 105 is important. And honestly, when you bring the samples, when you load a sample drying room with moist soil, it's going to struggle to get on over 95 degrees until all that soil dries out. And so it's most of the time it's less than 105, even though I said it's at 105.
Great, thank you. That was our final question. I'll let Jim go ahead and switch to our next slide. I thank everyone for their time today. Thank you so much, Jim. Thank you so much, Kevin, and everyone have a wonderful day.